Hello students. So today we are going to see cytokinesis in the chapter cell cycle and cell division. So what is cytokinesis and what are its type and in which type of cell we can see them? We'll discuss in this lecture. Now as we already know that cytokinesis is what? Cytokinesis is the division of cytoplasm. As we had seen in our earlier lecture, two types, that is karyokinesis and cytokinesis. So karyokinesis was division of nucleus and cytokinesis is division of cytoplasm. So cytokinesis can occur through two methods. As you can see on the screen, there are two types. One is the furrow method and the other is cell plate method. Now this furrow method can be seen in animal cell and cell plate method can be observed in plant cell. So let us quickly discuss how this furrow method takes place and how this cell plate method takes place. Now as you can see on the screen, the given diagram clearly shows how the contractile ring formation is taking place by the help of microfilaments and how the furrow method is proceeding towards cytokinesis to take place. Now as I said, in the equatorial region there is the formation of contractile ring which is formed by the microfilaments. Now microfilaments being contractile, what will it do? It will invaginate the plasma membrane and this will result in the formation of furrow. So the microfilaments, these are the microfilaments. These will not only help in the formation of the furrow, but it will also help the furrow to deepen up. As the furrow will deepen up, ultimately it will meet at the center and the cytokinesis will complete. So you can see gradually the furrow moves from the end towards the center like this. The furrow will move towards the center and it will look like as if it is cutting the cell into two equal halves. Now as I said, the furrow, it starts from the periphery or towards the end and it progress or move towards the center, and this is why this type of cytokinesis is called as centripetal cytokinesis. Keep this in mind that the furrow formation takes place which moves from outside towards inside, towards the center, and therefore this type of cytokinesis is called centripetal cytokinesis. Now let us talk about the second method, that is the cell plate method. In plant cells, small vesicles form by the Golgi bodies and they get collected at the center, or we can say at the equatorial region. Now these vesicles will fuse and after the fusion at the center they will form a thin film, and above the thin film and below the thin film what will form? There will be a formation of sheet, and these sheet will represent the plasma membrane of the two new daughter cells. So as I said, above the film and below the film the sheet is being formed, and as the cytokinesis will complete over here, these sheet will represent as the plasma membrane for the respective new daughter cells. Now look at the diagram and try to understand this. In the thin film there is accumulation of calcium and magnesium pectate. Now this calcium and magnesium pectate helps it to solidify, and when this thin film solidifies, the structure formed is called the cell plate. So basically this thin film, by accumulation of calcium and magnesium pectate, it will solidify, and this thin film will get converted into the cell plate, and this represents the middle lamella. Now as you can see in this diagram, the Golgi bodies, the vesicles that are formed by the Golgi bodies are accumulating at the center. So accumulation or arrangement of these vesicles formed by Golgi bodies is starting from center. Now as the cytokinesis proceed, what happens? New vesicles, or the vesicle starts to get attached at the ends of the already existing vesicles. So in this manner we can see that the arrangement of the vesicle is taking place from center towards the end. In previous method of cytokinesis, we saw that the furrow formation was taking place from periphery and it was proceeding towards the center. But here what we can see, that cytokinesis is proceeding towards the from the center towards the end. So here we can say that this type of cytokinesis is centrifugal type of cytokinesis. So this is centrifugal cytokinesis. Now let us talk about certain chemicals that are involved in cytokinesis, or we can say that help in inducing mitosis. First of all, these chemicals that induce mitosis are termed as mitogens. So when we talk about mitogens present in plants, these are auxins, gibberellins and cytokinins. These basically are plant growth hormones which are acting as mitogens in case of plant. And when we talk about animal, the mitogen is insulin. Now let us quickly talk about what are the different significances, or what are the different importances of mitosis. So first of all, we can see here the surface area or volume ratio. Now how we can understand this? Suppose if there is a small cell. Now this cell being small in size, its surface area is great. Okay. Now when cell is small but it is having a large surface area, there is an efficient exchange of materials within it. And as the cell size increase, the ratio decreases. Now as the cell size will increase, the surface area will decrease over here. Now if surface area will decrease, what will happen? Efficient exchange of material won't take place. So as the cell size increases, what happens? The cell quickly divides itself. The division of the cell takes place, therefore forming small sized daughter cells. Now this is involved in growth of multicellular organisms. Mitosis also helps in healing of wound. Whenever we get cut in any part of our body, what happens? Within few days it gets healed. So the cell is rapidly dividing and replacing the damaged cells. Now mitosis also helps in replacement of cells. For example, we can say when the red blood cells or white blood cells inside our body, their lifespan gets over, so they die out. So to replenish the loss of this RBC and WBC, what is happening? The formation of new RBC and WBC is taking place. So this is also taking place because of the mitosis. There are other certain examples like the outer layer of skin cells. Every time, every day, number of cells are being shed off from our body. So to replace or replenish the loss of these cells, what is happening? New cells are always being formed, and this formation of new cells are taking place by mitosis only. Now let us talk about meiosis, and I hope that the previous topic is very much clear to all of you. Now let us move on with this new topic, that is meiosis. Meiosis is also a kind of cell division, but it is different from mitosis. So let us know about this in detail. Now meiosis is a double division. This occurs in diploid cell. Now after the division, there is formation of 4 daughter cells. The number of chromosome is half in the daughter cells as that of the parent cell. Therefore, this meiosis is termed as reductional division. This term meiosis was given by the scientist named Farmer and Moore, and the details of this meiosis was studied by Benedin, Strasburger and Sultan. Now let us talk about the occurrence of meiosis. So as I have already told you that meiosis occurs in diploid cell, and the cells which undergo meiosis are called as myocyte. Now let us talk about meiosis in animals and certain lower plants. What these myocyte are in these animals and lower plants. So first let us talk about the gonads, that is testis and ovaries. Testis basically found in males and ovary basically found in females. Now in case of testis, the myocyte here is the spermatocyte, which is represented by 2n, that means it is the diploid. And when these spermatocyte, which are the myocytes, undergo meiosis, it gives rise to the sperm, that is represented as N, that means this is haploid. And when we are talking about the ovary, the myocyte over here is oocyte, which is again represented by 2n, that means it is diploid. And when these oocytes undergo meiosis, they produce large sized egg. Now large sized egg means that they are basically bigger as compared to the sperm. So the overall concept to understand over here is that how the gametes are formed. Haploid gametes are formed from diploid myocyte, whether it is the spermatocyte or it is the oocyte. Now let us talk about sporangia, basically found in the land plants. Now in case of sporangia, the sporocyte are the myocyte in this case. So we can see over here again microspore mother cell and megaspore mother cell. These two are acting as myocyte over here. Now both microspore mother cell and megaspore mother cell will again undergo meiosis, and the resultants will be from microspore mother cell, small spore microspore will get generated, which is again haploid, and from megaspore mother cell, large spores, megaspores will be generated, which is again haploid. Now when the meiosis occurs at the time of gamete formation, it is known as gametic meiosis. When the meiosis occurs at the time of spore formation, it is known as sporic meiosis. And when meiosis takes place in zygote, when we talk in case of lower plants like Chlamydomonas, Pyrogyra, etc., this kind of meiosis is termed as zygotic meiosis. Now let us talk about homologous chromosome. What are homologous chromosomes? Homologous chromosomes are pair of identical chromosomes with respect to size, position of genes, position of centromere. Now as you can see on the screen, for example, a diploid cell is taken where we can see presence of 4 chromosomes, that is 2 pairs of homologous chromosomes. Now in a typical chromosome there are 2 chromatids. As you can see, one chromatid, second chromatid. So in a typical chromosome there are 2 chromatids. Now here in the given figure, 1A has a homologous partner 1B, and 1A and 1B are the first copy of respective 2 chromosomes. The size is similar, centromere position is similar, with same position of genes. Now suppose if there is a gene which will indicate the color of eye, suppose over here, here and here. So whether we talk about the chromosome which is coming from paternal side or from maternal side, that means from male or female, the gene for a particular character has to be in the same position. So the same will be for the second copy, that is 2A and 2B. They will be homologous pair, that means 2A is the homologous partner of 2B. The size will be similar, centromere position will be similar, with same position of genes. So both the set of chromosome are similar with respect to different features, and therefore they are termed as homologous chromosome. So one set is contributed by male parent and the other is contributed by the female parent, and therefore it is termed as paternal set and maternal set respectively. Paternal set for male and maternal set for female. Now during meiosis 1, as it is the reductional division, 2 daughter cells are generated, which further undergoes meiosis 2 to generate 2 daughter cells respectively. Now when we are talking about the meiosis 2, that is equational division, the 2 daughter cells generated, the chromosomes in these daughter cells will replicate. It will not reduce, but the replication of this chromosome is taking place, and so we can say that meiosis 2 is equational division. When we'll study the different stages that includes in meiosis 1 as well as meiosis 2, everything will be clear to you all gradually.